Hi, this report is on long-read Amplicon sequencing, gene conversion, and spinal muscular atrophy. These are the disclosures. And spinal muscular atrophy, or SMA, is a disease of progressive spinal cord degeneration causing symmetric muscle weakness and atrophy. SMA is one of the most common autosomal recessive diseases worldwide, with an incidence in 1 in 10,000 live births. It's caused primarily by a bi-allelic loss of SMN1 exon 7 via deletion and or gene conversion of SMN1 to SMN2. Here we're going to show that bi-allelic SMN1 to 2 gene conversion presents with mild or completely absent disease phenotype. A consanguineous couple and 4 children presented to our lab. 2 children were asymptomatic, and the other 2 were diagnosed with mild type 3 SMA at ages 13-28 respectively. For each sample, we measured SMN1 exon 7 dosage using qPCR, and then we determined alleles using long-read Amplicon smart sequencing on the sequel system. We put it all together with segregation analysis, and here's what we got. qPCR results show something interesting is that we have, as expected, 2 affected children with 0 copies of SMN1, but we had 1 unaffected that had 0 copies as well, and 1 unaffected that had 2 copies. The parents predictably had carrier state of 1 copy of SMN1. Looking at the alleles in the long-read Amplicon data, we see that the parents are consanguineous. They have identical haplotypes, genotypes, for the normal SMN1 allele and their gene conversion SN1 to SMN2 allele, not picked up by the qPCR assay. The 3 children that are 0 copies are homozygous, identical for the gene conversion SM1 to SMN2, whereas the 1 unaffected, molecular unaffected child is homozygous for the normal SMN1 allele over here. So we found identical genotypes in the parents, 1 normal SMN chromosome, 1 gene conversion SMN chromosome, and the non-carrier unaffected child was homozygous for the normal SMN chromosome. The 2 affected children and 1 unaffected child were each homozygous for the gene converted SMN chromosome. And essentially what we're showing here is that although SMN1 to SMN2 gene conversion is common in the general population, it's quite difficult to detect using standard molecular techniques, unlike here. And here we show that the SMN gene conversion is readily detected by qPCR and long Amplicon sequencing. Our results suggest that bi-allelic loss of SMN1 by gene conversion is not necessarily indicative of disease.